lecture, and today we shall again discuss on biosafety, especially biosafety for the students, researchers in the laboratory, because it is very important. The safety first, whatever you do the research and you discover a big scientific issue or a solution of a problem, the most important issue is you have to keep you free from any danger. For example, now worldwide scientists have been doing research on COVID-19 uh, uh, disease causing virus SARS-CoV-2, and it is highly contagious, but even though scientists are doing the research, so how can they keep themselves free from the contamination and from the health hazard? When coronavirus first detected in Bangladesh in early March, within several weeks, we experienced or we witnessed infection of coronavirus uh, by many doctors, and we lost many doctors due to mainly, poor uh, personal protection equipments. And now you see that no doctor is infected. That means they are getting high quality personal protection equipments. And we had no experience of handling the coronavirus disease as well as the patients. So unfortunately, we lost many great doctors. Uh, Similarly, when you do research in the laboratory, biotechnology laboratory, you need to follow strict guidelines of the laboratory to keep you free from the uh, danger, contamination, as well as to keep free from others from the contamination. And it is a prerequisite to get the training on how you can keep yourself free from the contamination. Every laboratory in the world, research lab as well as labs in the university, no researcher can enter before receiving any training. But in our country, such type of routine training are not available in the universities. But in the developed country, they have routine training program for the entry of the laboratory, fires, hazardous materials, as well as some uh, equipments are being used. Let me start a slide. I'll show you some of the slides. Biosafety. Biosafety, already you know the definition of biosafety: to keep us safe from the biohazardous or potentially biohazardous modern biotechnological tools. But here I would like to discuss on the laboratory biosafety as well as biosafety in the research. Uh, Here, let us know one new term: biohazard. Uh, Biohazard, an agent of biological origin that has the capacity to produce deleterious effects on humans, animals, plants, and insects. Uh, they are called biohazardous substances or materials. These include microorganisms, toxins, and allergens derived from those organisms, and allergen and toxins derived from insects, animals, and plants. So in the laboratory, we use microorganisms, insects, animals, plants. Some of them are hazardous. They may produce toxin, they may produce allergens, and some of them may be, uh, for example, microorganisms even can infect human. So we have to be very careful about the bio hazardous substances in the laboratory so that they cannot harm the laboratory members, not only the person who is dealing the research. So how can yeah, we handle it? There is a chain of infection and risk assessment system. For example, reservoir of pathogen, where we preserve the pathogen. We preserve the pathogen in container, for example, petri dish or Eppendorf or something other containers, so that it cannot escape to the environment and affect the members. Then portal of escape. It may escape during your job, or when you are handling. It may escape to the environment, and then it can practical equipment also. The equipment you are using, for example, microscopy or other thing, the or even in the clean bench, if it is not biosafety level 2 or 3 or 4, then there is a chance of escaping the hazardous uh, organism to the environment of the laboratory. And then a transmission, route of entry or infectious dose. Uh, it may, this is another, you see, here it is written PPE, personal protection equipment, gloves, mask, or other, personal protection. If you are fail to use the right personal protection equipments, then it can get in touch with your body, and then it can cause the infection. In case of infection, I have no idea about the SARS-CoV-2 virus, how many particles are needed for causing human infection. But in case of most of the microorganisms, only one conidia or one cell is not, in most cases, not enough, because human immune system can protect it. So infection dose is required. Then other point is, just if you get in touch with the microorganism which is infectious, uh, human body has the protection mechanism. So susceptible host can easily be infected. If immunization is taken place for some researchers who are working on virus, usually immunization vaccine are taken, so that even accidentally they are exposed to the virus, virus cannot cause disease. And it takes some incubation period. For example, in case of SARS-CoV-2 virus, 5 to 7 days required for incubation in the human body before expressing the symptoms. So these are the steps which are very important to understand about the chain of infection by any uh, contagious organisms. Biosafety in microbiological and biomedical laboratories, there are some rule and responsibilities of the principal investigator or the researchers with respect to biosafety and animals. And the principal investigator is responsible for full compliance with the federal and state regulators. In our case, this is, I am telling about the US rules. They have the federal and state different regulations. Unfortunately, in case of biosafety of the laboratories, we have no straightforward uh, regulations in the country or the university. But in US, they have the specific and well-defined federal and state regulations. NIH, National Institute of Health, and CDC, guidelines. They have the guidelines. How can you handle them? And institutional requirements for research involving biohazardous materials. I told you this is most important. Institutional requirements for research involving biohazardous materials. Every institution should have, some requirements, guidelines, or rules for protecting the researchers who are involved in research with biohazardous materials. So, uh, principle of biosafety, these are common. This is why commonly all the laboratories all over the world should maintain these principles. Containment is very important. Where you are, preserving your uh, biohazardous materials. This is very important so that it cannot escape from the container. For example, if any poisonous or pesticide or insecticide containing any bottle or container, what we do, we preserve in a place so that the children and other person cannot easily get the access, so that they can abuse it. So the purpose of containment is to reduce or eliminate exposure of laboratory workers, other persons, and the environment. So laboratory workers, other persons, and the environment, we must keep them free. Safe methods for managing infectious agents in the laboratory. Environment, the location where the agent being handled and maintained or stored. So these are very important principle. Every laboratory should be careful and maintain. Mode of infection, how the infection is taken place. This is important. Infections are preceded by overt personal accidents, which include inoculation, resulting from pricking, jabbing, and cutting the skin with contaminated instruments such as hypodermic needles, scalpels, glass wires from animal bites or scratches. If someone has this type of injury, then there is high chance of contamination by the microorganisms, because most of the microorganisms they are protected by our skin. Uh, if they find any wound or any entry point, then they can easily infect the human. Ingestions, somehow if inhale or ingestion taken place, then it is dangerous, resulting from mouth pipetting, sometimes mouth pipetting, eating, drinking, or smoking. In the laboratory, it is strictly prohibited to eat anything, drink anything, or do smoking. All over the world, the biology laboratory, especially biotechnological laboratory, you are not allowed to eat, drink, and smoking, because environment may contain hazardous substances there, and it may cause ingestion, unintentional ingestion, and result serious consequences. Splashing into the face and eyes when you are handling the organism or hazardous substances, splashing may take place. Spillage and direct contact. Sometimes, when people work in the clean bench, and handle the hazardous substances as well as organisms, spillage, and direct contact may happen. So there should be a very careful. This is why goggle and mask are being suggested, and obviously the hand gloves. Infections not preceded by personal accident, for example, aerosol. It is not a personal accident, but aerosol, droplets, or fomites. In case of COVID-19, we use mask. A mask is essential for the person who is attacked by the COVID-19 uh, disease causing virus, that is SARS-CoV-2, or those who have no disease, because to protect themselves, mask can protect a certain percentage, a high percentage of the, protect from the aerosol droplets and fomites. These are speculated from Pike's scientist in 1976 data to be responsible for up to 82% of all laboratory acquired infections are caused by the unintended infections not preceded by personal accident. Aerosol are a cloud of very small liquid droplets produced whenever energy is applied to a liquid or such liquid is allowed to escape into the environment. Centrifugation is a good source. So when you do centrifugation, there is a high chance of aerosolized plasmid and other, microorganism, and unintentionally, or without personal accident, you may be a victim. There are many regulations in place to forestall the problem of laboratory acquired infections. Um, for example, responsibility for compliance with regulations, primary to the principal investigator, secondary with the laboratory staff. So everyone need to, abide by the rules, strictly follow the rules. If you have no rules in the laboratory, you should check in the internet, because every biological laboratory all over the world and country have the rules. So the rules and regulations we have to follow, from principal investigator to the laboratory staff, even a cleaner, to keep themselves free from the hazard. In general, a large number of organisms that would ordinarily be innocuous can be infective in immune compromised persons. This is very important. For example, you are using a fungus or a bacteria which is associated with plant or fishes. Naturally, they are not infectious for the human, but if you are immunocompromised, this is very important point. Immunocompromised, sometimes they can, infect you. So, weak pathogen, or maybe are not direct pathogen, but if you are immunocompromised, or if there is any wounds or any entry point in your body, microorganism can proliferate and can cause hazardous situation. Therefore, additional and more stringent measures must be established by the PI in an effort to prevent the occurrence of lab-acquired infections in such individuals. So, immunocompromised, what does it mean? When you are under severe stress, or when you are maybe suffering from other diseases. In that case, your immune system is compromised, means weak. So this weakness, immune system is the guard and real protector of any living organism from other external, enemies like microorganisms. So in that case, immunocompromised uh, persons are prone to infect by the even weak organism. Primary containment, protection from exposure to infectious agents, is provided by both good microbiological technique and the use of appropriate safety equipment. So, primary containment and secondary containment. For example, you put something in a bottle, and then it should be put in a place where you can lock it in a house cabinet or like that. Secondary containment, protection of the environment external to the laboratory from exposure to the infection materials like facility design, operational practices. So laboratory design is very, very crucial in that case. So three elements of containment include laboratory practice and techniques. So every laboratory should have, standard practice and technique, and safety equipment, Bunsen burner or bacteria incinerators, cases and other barriers. Facility design, the risk assessment of the work to be done with a specific agent will determine the appropriate combination of these elements. For example, clean bench or laminar airflow cabinet. There are two types of laminar airflow cabinet. Some of them are biosafety cabinet. For example, we have a biosafety level 2 cabinet or 3 cabinet. Those are safe for using any hazardous materials. For example, for coronavirus, the people who are doing real-time PCR to detect the presence of coronavirus in the sample from the suspected patient or human. They do in the biosafety level 2 or 3 cabinets inside them, because no chance to get out or contaminate the environment or air of the laboratory. So this is very important. Otherwise, the person who is involved in diagnosis, they will be contaminated. Laboratory practice and techniques, I would like to add more point. Standard microbiological practices and techniques should be followed. Persons working with infectious agents or potentially infected materials must be aware of the potential hazards and must be trained and proficient in practices and techniques required for handling such materials safely. So, first you have to know which materials you are using and what are the potential hazards associated with that material, and then you have to follow the standard practice as well as techniques for handling them. The director or person in charge of the laboratory is responsible for providing or arranging for appropriate training of the personnel. In the developed country, I told you, anyone who want to work in the laboratory, any biological laboratory, uh, he or she must appear or attend a training and exam. Otherwise, you will not be allowed. Even when I was a mature scientist, but when I was at West Virginia University in 2017, very recently, uh, I was not allowed to enter into the laboratory before receiving the training from their central training division. So, a scientist trained and knowledgeable in appropriate laboratory techniques, safety procedures, and hazards associated with handling infectious agents must direct laboratory activities. Because who is the director or laboratory manager must know all the procedures and safety issues. Each laboratory should develop or adopt a biosafety or operational manual which identifies the hazards and will or may be encountered, and which specifies practices and procedures designed to minimize and eliminate the risk. All personnel should be advised of special hazards and should be required to read and follow the required practices and procedures. Primary requirement of entry any biotechnology or biological laboratory, you must wear the gown, that means apron. Each individual and never use the apron outside the laboratory. Nowadays, even in Bangladesh, some funny thing we can see. Some medical student, they use their gown or apron outside the laboratory. It is like a fashion, but it should not be a fashion. It is, some personal protective equipment. Each individual in the laboratory should acknowledge that they have been advised and briefed as to the laboratory's plan and to the risk that they will be exposed. There is no such thing as zero risk. So always there is a risk. So laboratory containment levels for biological research involving potential hazards include risk assessment, recommended biosafety levels for infectious agents and infected animals, and agent summary statement, and these should be included in the manual, laboratory manuals also. So guidelines for research involving recombinant DNA molecules or the in at the, any other materials which are hazardous. For example, recombinant DNA, molecules, they may contain plasmid and there is a high chance of contamination. So you have to follow the guidelines. If you have no guidelines, you can follow the NIH guidelines for research involving the recombinant DNA molecules. So many countries follow these guidelines, and it is worldwide acceptable, and we can make our own one also. So guidelines for research involving recombinant DNA molecules, there are several risk group agents, for example, RG1, RG2, RG3, RG4. So all are not the same risk group. So you have to understand which risk group you are handling. So, physical guidelines for research involving recombinant DNA molecules, the most important is physical and biological containment for recombinant DNA research involving animals. Because, animals are used as model organism for doing any research related to the animal genetic engineering. So you can see, depending on the level of the hazard, you can use biosafety level 1, biosafety level 2, biosafety level 3, and biosafety level 4. So there are 4 categories depending on the level of or associated hazards. And disposal of the animals is very important after slaughtering or doing the experiment, where you dispose the materials. And even in our laboratory, when we do PCR and other um, uh, uh, job where hazardous materials are being used, some of them are carcinogenic. So in that case, disposal of the waste is very important. In Bangladesh, we have a very big problem. The amid coronavirus, the Dhaka city or other cities, people are using mask and different personal protecting equipments like gloves and others. Even hospital waste, they are mixing up with the normal waste, and municipal workers, they are not separating, and no yellow bin or bags are used to uh, carefully handle those coronavirus contaminated waste, and it is a big problem. And recently, there was a competition from the Geography and Environment Society on essay competition. And one essay drew my attention from Bongabandhu Agriculture University, an undergrad student from veterinary and animal science, Mr. Faisal. He became the champion in writing the essay, and his essay, the topic was hazardous medical challenges associated with hazardous medical waste in amid COVID-19, and he focused the international rules and regulations, national rules and regulations, and real practice we are not following. As a result, the coronavirus contaminated as personal protecting gloves, masks, and other things, polluting the other medical waste, and that virus may be spread to the environment as well as sewage water, and it may create a new problem of new infection through the environmental contamination. And I was so impressed to read his, uh, essay, and I invited him to, discuss with me, and we had a very nice discussion. And I am trying now to guide him how to write it in a scientific way so that he can publish it in an international journal. So, as a student of environmental biotech technology and biosafety, please think carefully and seriously how you can uh, 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 be involved in such type of review on associated with the COVID-19 hazards, because it is a big hazard, not only confined in the laboratory, it is now open to the country. So our whole country is now under a threat. Anybody in any time may be contaminated. Many people are contaminated without any symptom. They may contaminate unconsciously or even without notice to the healthy person. Yesterday, I knocked one of my friend who is the chief editor of, Dhaka Tribune, for publishing one of our press release. I came to know that his all family members are infected by SARS-CoV-2. So it is a really a terrible situation. As a young environmental biotechnologist and specialist in biosafety, please think you may also focus some issue uh, uh, which is very important for tackling this highly contagious virus, and even you can publish in newspaper or in the journal. Guidelines for research involving recombinant DNA molecules was exempt. Some of the those that are not in organism or virus, some of them are not hazardous. Those that are not in organisms or viruses, those that consist entirely of DNA segments from a single non-chromosomal or viral DNA source, though one or more of the segments may be synthetic. Those that consist of entirely DNA from a prokaryotic host, including its indigenous plasmids or viruses when propagated in that host, or when transferred to another host by well-established physiological means. Um, those that consist entirely of DNA from a eukaryotic host, including its indigenous chloroplast, mitochondria, or plasmid, when propagated in that host. So that can be, exempt from the strict regulation. Bottom line is, PCR, gel running, these are also not so problematic, but depending on the materials you are using for the doing the PCR or gel running is very important. So biosafety guidelines. I had a visit to a medical university of South Carolina. I got I attended such a training there. How to determine the relative biosafety risk associated with the planned experiment. This is a question after the training, and there are several specific questions. So specific requirements for biosafety level 1. For biosafety level 1 to 3, you need PPE, personal protection equipment. That means you need an apron or gown, and you need gloves, you need mask, you need goggles to protect yourself. Biological safety cabinets, you need, that means a laminar airflow cabinet. Risk associated with centrifugation and proper protocols. So uh, you should maintain it. And by principles of biosafety levels, what are BCL1 to BCL4? Biosafety level 1 means agents not known to cause disease. For example, if you use a root from a horticultural plant to dissect or to get the beneficial fungus or bacteria from it, it is, you can handle or consider it biosafety level 1 because that agent not known to cause disease. Biosafety level 2, agents associated with human disease. For example, COVID-19 causing a virus SARS-CoV-2. It is an agent associated with human disease, and you must minimum follow the strict guidelines of biosafety level 2, but it is very contagious. This is why we suggest to use such thing in biosafety level 3. Biosafety level 3, indigenous or exotic agents associated with human disease and with potential for aerosol transmission. So in case of coronavirus, pathogen, that is coronavirus 2, SARS-CoV-2, it is potential for aerosol transmission. So biosafety level 3 is highly recommended. BSL4, dangerous exotic agents of life threatening in nature. Even coronavirus 2, that is SARS-CoV-2, you can consider biosafety level 4. So better to use at least biosafety level 3 or 4 facilities or the principles. So biosafety level 1, suitable for work involving well-characterized agent, not known to cause disease in healthy adult humans or minimal potential hazard to laboratory personnel and the environment. These are biosafety level 1. And for biosafety level 1 to maintain it, and the requirements are laboratories must have doors, a sink for hand washing, work surfaces easily clean, bench tops are impervious to water, sturdy furniture, and windows fitted with fly screens. So these are the requirements for biosafety level 1. Biosafety level 1 standard, microbiological practice use mechanical pipetting and not using mouth, so that and you see the mask and apron, not mask, gloves and aprons are being used. So this photograph gives you the requirement for or practice of standard microbiological practice for biosafety level 1. And you must, after the job, you must wash your hand by using the soap or detergent. Liquid soap is the best. And minimize splashes and aerosols. So when you do the job, decontaminate work surfaces daily, decontaminate wastes. Yes, this is most important. How do you decontaminate waste? You can do autoclave, maintain insects and rodent control program, because some people are working with and uh, materials which may be infected or may be loved by the insect or rodent. So you have to maintain it in the laboratory so that the materials are not attacked by the insect or rodent. And protective clothing like lab coat and gloves must be used, and personal protective equipments, face protection, eye protection. Sometimes you need to use the goggles and even mask, uh, depending on the the risk in the associated with the materials you are using. Biosafety. Let me discuss on biosafety level 2. So in facility design, you should have secondary barriers. Requirements: laboratories have lockable doors, sink for hand washing, work surface easily clean, bench tops are impervious to water, that means waterproof, sturdy furniture. So these are essential. And uh, requirements: biological safety cabinets installed as needed, adequate illumination, that means light, eye wash readily available, air flows into lab without recirculation to non-lab areas, windows fitted with fly screens, restricted access when work in progress. When you are doing the work, the laboratory door should not be suddenly open, because when you open the door, the big airflow can be happen, and then it may cause hazard. For example, you are using some materials in the cabinet, so your wave can take out the materials and pollute the environment of the laboratory. So BSL1 facilities plus autoclave available. Autoclave must be available so that you can, uh, you know, uh, sterilize the materials after use. Eye wash station needs to be available so that you can wash your eye if there is any uh, splash on your eye. Use biosafety cabinets class 2. Yes, we have class 2 facilities for work with infectious agents involving aerosol splashes, large volumes, and high concentration. And here you can see the biosafety level cabinet equipment layout, how you can put the things inside the cabinet, and cabinet should be like that, and you have already seen. And in addition to BSL1, use biosafety cabinet class 2 for work with infectious agents involving aerosol splashes, large volumes, and high concentration. Yes, already I discussed this. uh, Biosafety cabinet, how can you use it? You can see the door, how much you can open it, just so that you can enter your hand and not open more than it would be problematic for your mouth or eyes and others. Use biosafety cabinet for work with the infectious agent like aerosol, splashes, large volume, high concentration. Already I mentioned it. Yes. Vacuum lines protected with liquid disinfectant traps for HEPA filters, because otherwise HEPA filters, if it is used in the cabinet. So if it is you know not timely replaced, then efficiency may be reduced, and it can cause the coming out of the hazardous microorganisms from the cabinet. Supervision supervisor is a competent scientist with increased responsibility. So regular supervision is necessary, limits access if immunocompromised, restricts access to immunized, Lab personnel aware of potential hazard, proficient in practice and technique. So these special practices should be carried out. And another is needles and sharp precautions. Use sharp containers, don't break, bend, or resheath or reuse syringes or needles. These are very problematic. And supervision, already I mentioned, yes. Don't place needles or sharps in office waste container because that should be in different containers so that not mix up with the normal laboratory waste. Yes. Needles and sharp precautions, do not touch broken glass with hands, and then it may cause a problem. And policies and procedures for entry. Biohazard warning sign, biosafety manual specific to the lab, training with annual updates. These are lots of things you need to do. So supervision regular, I mentioned, yeah. Use leak-proof transport containers when you move materials from one place to another. And immunization, in some cases, vaccination is necessary for handling some of the materials. Baseline serum samples. Now we have no coronavirus vaccine. Otherwise, the people who are working or doing research with SARS-CoV-2 or who are working in the diagnostic lab, they should take vaccine first. But we have no available vaccine for SARS-CoV-2. Decontaminate work surface, report spills and accidents immediately. No animals in the laboratory. So these are up to the biosafety level 2. Human blood, blood products, body fluids, tissue are listed as potentially hazardous biological materials. Biosafety level 2 practices and procedures must be followed when handling blood, blood products, body fluids, tissues, and under no circumstances shall anyone work with cells derived from themselves or from first degree relatives, since the host immune system may not provide adequate protection. So there is a green book for greater detail, green book available in the internet. So this is all about up to biosafety level 2, and in case of cell culture, obviously you can follow. So I would like to stop today here and discuss with you more in detail. So now you can ask some question because this is very, very important for the life. You are going to start your MS research. During the research, these useful informations would save your lab members as well as the environment. And later we shall discuss on biosafety level 3 and 4. So any question from your side?
Sir, uh,
Yes.
sir, we don't have biosafety level 3 or 4 laboratories in our country.
Uh,
Do you,
we have IEDCR as well as ICDDRB. They have possibly, I didn't visit there, but they have possibly biosafety level 3 labs. Because cholera pathogen is very contagious pathogen, and ICDDRB they are doing the research, and IUDCR I heard they have the biosafety level 3. facilities. But other universities or research institutes so far are no biosafety level 3 or 4 laboratories. Uh, and in case of agriculture, people do not maintain any restriction. But in case of biotechnology and genetic engineering, as a researcher, I must train the students before entering the laboratory. This is why I included this topic, which is very relevant to your course, and you are lucky because you are getting first-hand, training. Any question? Everyone needs to ask at least one question because you are only, small number of students. So if you ask question, it is also an art of learning something. Asking a question is really the expression of your talents as well as your intellectual capacity. So this training in the classroom, you can learn and do practice. In the practical life, it is very useful. Whenever in your job or even a conference and a meeting, if someone can ask an intelligent question, people attention you can draw, and it is also associated with the career of any person. So uh, try to ask as many as questions to me because I love question, any type of question. Uh, uh, I can try to give you answer if the answer is ready in my mind. Otherwise, I can refer you to the literature. So, Sumaya Talukdar,
Yes, sir. Assalamualaikum, sir.
Walaikum Salam. And you can do practice of communicating in English in this classroom, and it would improve your skill very much. And it is not only essential for studying abroad, it is essential and important for our society also. Society value it.
Sir, in our laboratory, we have sinks for washing hand, and we also washing their different laboratories waste. Sir, isn't this take part to contaminate our groundwater?
Yes, groundwater. Groundwater, our water in our campus, water is very good quality because we have own deep tube well, but the water in Dhaka city, Wasa water is not good. But in our case, whenever we use any microorganism in the laboratory, we must, for example, in the petri dish or in the flask, we should not throw them in the basin uh, and contaminate the, sewage system as well as the environment of the university. It is ideal, and I always suggest to go for autoclaving because we have the autoclave. So you can autoclave and kill all the bacteria and destroy the DNA plasmid, and then it is safe, and you can release them to the basin, and uh, then it will be safe.
Thank you, sir.
Sir, is it possible to is it possible to decontaminate those waters before releasing that in the groundwater, like any type of plant that can be decontaminate those waters?
A plant actually, uh, we are using mostly the crop plants as well as weeds to collect the microorganisms or the beneficial microorganisms or pathogenic microorganisms. Usually, they are biosafety level 1. So in that case, autoclaving is not necessary, but after the use, the materials should be, they are like, household kitchen waste like. So no problem to uh, put into the bin of common waste. But in our laboratory, unfortunately, we have no yellow bin. Uh, we maintain some other mechanism, but no standard yellow bin for uh, how to say to preserve the hazardous waste. And we have no incineration in the modern world. Incineration is used to using to destroy all the hazardous waste. For example, 3000 degrees Celsius. For example, medical waste. Medical waste should be incinerated at 3000 or more or higher temperatures, so that completely you can destroy all the possible organisms present there and keep your environment safe. In Bangladesh, very limited number of incineration facilities are available in maybe in the medical college or like the hospital. But in case of university laboratories, um, our system of maintaining the hazardous waste, management of hazardous waste, still faulty. So we have to work hard to find the way. And biotechnology, is a new area of research. We are still at the infant, so we have to do a lot of work. And other departments in agricultural universities, they are not dealing with uh, hazardous substances, so their waste can be, composted or like that, and not so problematic. But my suggestion is, when you work in our laboratory, you must be very careful and remember all the story I am telling you, so that you can keep yourself free from any contamination or hazard, biohazard. Thank you, Fatima. And next,
Sir, Assalamu alaikum. Jinan sir.
Jinan, please.
Sir, air conditioned water, I mean the water which is generated from AC is very much acidic, but it can be converted into drinking water. But if we want to use it in the crop field, so what kind of biosafety measures we can take or what we can follow?
I have no idea about, air condition, air leakage, water leakage from the air conditioner. Sometimes, if air conditioner is defective, then air drops are coming out, and that may be acidic. I am not sure. But those water, if it is acidic or contains some other hazardous materials, uh, uh, the one way we need to, autoclave it before disposing to the environment. And we need to be careful, but I have no idea how much water is coming to the environment from the refrigerator. And it is not in the inside the laboratories. If we see a dropping of water from the uh, uh, air conditioner, then we repair it quickly, so that not contaminate the laboratory or cause problem.
Okay, sir. Thank you, sir.
Welcome. Any other? Jahid?
So I don't have any question actually. I have a question. Sir, can I, sir, ask you a question outside the today's topic, sir? Regarding yesterday's topic, a question arose in my mind.
What kind of question?
Sir, you yesterday I asked about conventional breeding and sir, using biotechnology for breeding hybrid crops. Sir, the question arose that why didn't I use the conventional breeding technique if I want to transfer gene rice from rice? Uh,
Yeah, gene rice from rice, you can do conventional breeding as they are compatible for crossing. So
sir,
in that case, you cannot specifically control only the transfer of your target gene. Lots of gene will be transferred, but there is an efficiency, maybe 5 to 12 years is necessary to transfer the specific gene. In that case, no need to
sir,
biosafety guideline.
I can't hear you.
You cannot hear me?
Sir, now I can hear you. Sir, my question is, sir, if using biotechnology is more efficient and more specific, and in biotechnology the sources of genes are unlimited. So why aren't we adopting, sir, biotechnological method like we are adopting classical breeding method?
Pardon, I cannot hear your question. Yes, it is true that the source of gene is no matter for a biotechnologist, because if you can characterize a gene and clone it, then you can transfer it into any organism, any cell. There is no limit. And if it is from rice to rice, for example, CRISPR-Cas genome editing, if you do it, it is called, cisgenic, and it is people's sensitivity to the that product would not be like, transgenic. So it should be definitely it's obviously safe, but it should be. But I'm not sure exactly, because our policymaker always consider the process, not the product. So process. If we use the transgenic approach, or if we use the gene editing approach, some of the countries, they are very strict about to follow the strict guidelines. Do not matter whether it is from the same species or not. But I think if it is from the same species, it makes no sense to follow, all the strict guidelines. And actually, guidelines, that biosafety guidelines that I described, they are just to satisfy the mainly to satisfy the policymaker and environmentalist. Um, Most of them are, wastage of time. For example, a pharmaceutical drug, antibiotic, when we are getting it from discovered in USA, and it has been using all over the country, all the countries in the world. And in US, when it was discovered, they followed strict biosafety guidelines, and it is all. And no other country, do the same thing before using for their own people. But in case of um, genetically engineered plants, still people are, foolish. If it is found safe in USA, still Bangladesh is doing from A to Z biosafety guideline, for example, for the BT brinjal or like that. It makes actually no sense as a scientist, but to satisfy the regulators, this is the practice people are doing almost all over the countries, all of the countries. Is it make sense, your question? Answer of your question, Tahir?
Yes, sir. Yes,
Any
sir.
more
Sir, I
question?
think yes, sir. I think it would be more time saving if we use modern technologies. My thought is that from that thought, the question arose in my mind.
Mm-hmm. Modern biotechnology, do you think that every country should, for example, BT brinjal, if India want to introduce BT brinjal, they should also do from A to Z biosafety guideline? Because it is already been established. Thousands of people have already eaten the BT brinjal in Bangladesh. But if you go for India, they will ask, or Pakistan, they will ask A to Z, biosafety guidelines. Does it make sense?
I think it's many of the biosafety rules doesn't actually make sense.
Yeah,
So they are not logical. I think they are mainly, sir, like a conspiracy or something that genetically modified organism will harm like this way or that way. Mainly those which are like stories.
exactly. Any more question?
No, sir.
Assalamualaikum, sir.
I have another question. Sir, does the biosafety level vary from country to country, or it is fixed for every all the countries?
Biosafety, after Cartagena Protocol and global biodiversity regulations, biosafety guidelines all over the world follow some basic, guidelines or principles. But every country has its own biosafety guideline. For example, Bangladesh. So I suggest you to browse internet to find the latest biosafety guidelines for Bangladesh. So country to country there are slight variation, but not so big variation. Yes, every country has its own, regulation, own guidelines passed by the, regulatory authorities. Uh, Ruhul Amin Rana, you
Sir.
want to say something?
Yes, sir. Yes, sir.
Yeah.
Sir, what is the difference between immune system and immunocompromised?
What is the difference between
Immune system.
immune system and?
Sorry, immune system and immunocompromised.
Oh, okay. Immune system, all of us, human being, or, mammals, even a plant have its immune system. Immune system means defense system. For example, in Bangladesh, we have Army, Navy, and Air Force. They are the immune system of Bangladesh. They guard Bangladesh so that other enemy cannot capture our sovereignty, our independence. Similarly, in the organism like human, we have the immune system. For example, if any toxic substances or any microorganisms somehow get uh, into our body, then our body can sense them. There is the sensing mechanism, and they show some response. Response means they produce some antibody. Antibody means protein to tackle the the enemy. For example, a microorganism, and tackle the microorganism. There is T cell and other white blood cells. They act together and in a concerted way to kill and detoxify the invaders. Uh, it is called immune system. And a healthy young man, woman has a strong immune system. But when you are in stressful situation, when you are already uh, sick by other pathogen, that means your immune system is weak, and they are called immunocompromised. So in that case, even a weak pathogen can collapse you. So in most cases, whenever a person is attacked by any pathogen, for example, SARS-CoV-2, the pathogen of COVID-19, when infect a person, some persons, they become very weak, and respiratory problem and other organ failure we can see, and finally result the death. And we recently collected the samples from the different patients and did the genome sequencing, metagenome sequencing. That means next generation sequencing of the sample from different patients, and we found that when COVID attacked, many other microorganisms also participate, and they are usually not, severe pathogen, but they participate and jointly, collapse the patient because if COVID-19 already make the patient immunocompromised, then others can easily invade and pass through the antibody, that is the defense system. So this is the case whenever any plants or any organisms are attacked by a virulent pathogen. Virulent means which have the ability to overcome the defense system of the host. In that case, secondary, tertiary, even saprophytic pathogens participate to get the food from the, damaging tissue by the primary, pathogen. And it is very common. In case of wheat blast, we found the wheat blast infected spikes contain lots of other weak pathogen, fungi and bacteria that ultimately or rapidly destroy the spike as well as the grain that initially or primarily infected by the Magna Porte orizi triticum pathogen, which is a virulent pathogen for collapsing the wheat. Clear?
Yes, sir.
Okay. Do anyone have any question? If no question, then I shall end today's lecture with a special thanks to all of you. And I wish all of you safe stay and healthy stay so that we can continue our life, in a good way to attend the class and see you in the next class.